All right, hi everyone. Today we're going to be going over nervous system anatomy. So let's get started. All right, so here is the info that you are all probably aware of. In case you haven't heard, we are now officially a 501c3 nonprofit. So thank you for supporting us, and we hope we can continue supporting you in the future. Any prep course questions before we start? All right, if not, we can go ahead. Feel free to drop them in the chat at any time. So here's our session agenda for today. We're going to go over some of the homework materials that we recommended, and then do some content review over high yield concepts, and then a practice passage together. So to start, any questions over the homework or concepts or anything for this class over nervous system anatomy? Okay, if not, I can move on to a couple of problems that I pulled that I think might be useful. And just going forward, like if you have anything that you want to put in the chat, feel free to just throw an asterisk in there so I know that you're currently typing, so I know to wait for you. Okay, so here is our first question from the homework. What term describes how a neuron downregulates its responsiveness over time to a constant stimulus? So the answer to this question was adaptation, assuming that you had a chance to look over beforehand. The reason I wanted to highlight this question is because I think that something that people might get confused with is adaptation and habituation. So habituation is actually referring to the behavioral response that occurs, or a decreased behavioral response that occurs through repeated exposure. Dishabituation is kind of the opposite, where you no longer have that, you no longer have desensitization to whatever stimulus we're talking about. And then adaptation is specifically referring to the neural processes behind this. So what's going on in your brain and in your nervous system, rather than the behavioral side of things. Any questions over that? All right, if not, we can move on. Here's another question that I wanted to highlight. This is just over something called the magnocellular pathway. This actually, I feel, is a low yield concept, but I figured since we're doing a class anyways, the way that I usually came across a lot of random low yield stuff was just talking and studying with other people and doing random practice problems. And so I figured, if something sticks in your brain that's low yield, may as well bring it up. So this is asking about attributes of the magnocellular pathway. The answer here is high temporal resolution. So what the magnocellular pathway is it sends information about movement to your brain. Since most of the retinal ganglia that exist in your eye exist outside of the fovea or the focus area. And so it gives you movement or information about movement of really large, fast things. So that's what we would refer to as low spatial frequency and high temporal frequency. Any questions over this? All right, and then one more question from the homework that I wanted to mention. Again, I think just personally, this is a low yield concept. I had not even come across this until I did the Khan Academy myself. So when a cup starts to slip from one's hand, it makes quick vibrations which are felt through the hand. Which receptor that is important in perceiving vibration is being triggered? So obviously, you're going to assume it's a mechanoreceptor, and all the answer choices point to that. But then it's asking you to choose between slow adapting and fast adapting, and then even further between the types of fast adapting and slow adapting that exist. So hopefully you're familiar with slow adapting, fast adapting. If not, we're going to go over it in content review, so no worries there. But this is actually going to be a fast adapting mechanoreceptor. And the reason for this is because, as you study this nervous system anatomy, you'll learn that there are different situations where your body needs to have a slow adapting versus fast adapting response. In this case, if you're just feeling something with quick vibrations slipping through your hand, and you can even think about this from a personal perspective, like when you have something moving in your hand, you don't feel that sensation after it's left your hand usually. And so that's fast adapting. And then this just is something like a detail knowledge, whether it's Meissner or Pacinian, which, like I said, I just wanted to include this because I myself had not come across this before. So in case you're someone who picks up random facts, maybe it's helpful to you. So that was all for the homework review. Again, any questions over anything that we've covered so far or anything that you came across during your pre-class preparation? Okay, if not, we can move on to talking about the content. So quick overview, these are just some functions of the nervous system. I'm sure all of you are familiar with. And these are pretty easy to Google, so I won't spend too much time on them. But your nervous system is important for a lot of different functions, obviously. So there are some different divisions of the nervous system, and I think this is pretty important to understand just from an overview perspective. So your central nervous system and your peripheral nervous system are the two main divisions that you have. Your central nervous system consists of your brain and spinal cord, and then your peripheral nervous system is cranial and spinal nerves. So those are all the peripheral nerves that are extending from your brain and spinal cord. And then within your peripheral nervous system, you'll have two more divisions, your sensory division and your motor division. And your sensory division is going to be afferent. So that means that you have nerves sending signals to the brain and spinal cord. And then your motor division, which is efferent, so signals that are exiting or leaving. And then your motor division is further divided up into somatic and autonomic nervous systems. So your somatic responses are your voluntary ones, so just moving a muscle. And then your autonomic are involuntary, so smooth muscles, so stuff like heart rate, blood pressure, digestion, all of that. So we're going to take a closer look at the spinal cord first. So this is an image of the spinal cord and the parts that you should be familiar with. Oftentimes, I think you'll come across things about the dorsal horn, dorsal root ganglion, ventral horn, all this kind of stuff. So knowing the different functions and what's present in those locations is important. So your dorsal and ventral horn, this like darker stuff on the inside, this is your gray matter. And gray matter is what contains your sensory and your motor nuclei. And then you have your white matter surrounding all of that. And that's just tracts of axons which carry information to and from the brain. One thing that I want to make as a distinction here, so you see the words nuclei and ganglion come up a lot, and I think it's important to know the difference. So nuclei specifically are referring to clusters of nerve cell bodies in the central nervous system, and ganglia are in the peripheral nervous system. Similarly, there again, clusters of nerve cells that all have the same function, but ganglia is peripheral nervous system, nuclei are central nervous system. Any questions over that? Okay. And again, just if you have questions and you're typing something out because you don't feel comfortable unmuting, that's okay. Just, drop an asterisk or something in the chat so I know to wait for you. Okay, so now we're going to look at spinal reflex arcs. So these can be autonomic or somatic reflex arcs, and there's two main types. They can be monosynaptic or polysynaptic. So an example of monosynaptic reflex arcs is a stretch reflex. Sometimes you might come across it called like a deep tendon reflex or something like that. And so what happens here is that you have a stimulus that triggers sensory information to be sent to, or to be basically sensory information travels through a sensory neuron. And then it goes straight to a motor neuron that evokes a response. So this is called monosynaptic from the name because there's only one synapse in this reflex. You're going straight from sensory to motor. An example of this is like the knee-jerk reflex. Then you have your polysynaptic reflex arc, which is also sometimes called a withdrawal reflex, or that's an example of one. The only difference here is that you will have multiple neurons involved. And so you have your sensory neuron, then you have one or more interneurons, then you have your motor neuron, and then you have a response. And so those interneurons are considered your integrating center. And in both cases, the information is passing through your spinal cord, but the difference is that there just happens to be some interneurons involved with your withdrawal reflex. In both cases, even though only the spinal cord is involved, it's important to note that information can still be sent to your brain. So your brain might not be involved in the actual reflex, but you might still get sensory information like you touch something hot. It's just not involved in the actual motion of removing your hand from the hot object you're touching. Any questions over that? Okay. So let's do a quick knowledge check. So which of the following would be most likely to be damaged in a patient that presents with weakness in their arm? I'll give you all a minute or two to think about that. And if you're comfortable, please drop something in the chat. It's okay to take a guess. Okay, so for this question, the correct answer is B, efferent neurons in the peripheral nervous system. So efferent neurons, if you'll remember, are the ones that are carrying information away from the brain. They're your motor neurons that are stimulating muscle contraction. So your peripheral nervous system is all of the neurons that would be innervating your arm. And so that's why B is the most likely answer. Afferent neurons, you can rule out A and C because sensory neurons are afferent. You can rule out because, again, they carry information to the brain. So they wouldn't likely be damaged or causing weakness in the arm at least. And then interneurons, those are involved in reflex arcs like we just talked about. So they don't really apply here. Any questions over that? Okay. Moving on. All right, so this is just the diagram that I like from one of my textbooks over parts of the brain. I won't go too far in depth here because I think probably a lot of you are familiar with this and it's not too difficult to look up, but anybody have any questions or want to go over anything here specifically? Okay, I'm not seeing any takers, so we can keep going. So let's see. All right, so this is some stuff over the autonomic nervous system. Don't be scared off by this diagram. I just included it because I think it's really helpful in terms of talking about the different responses that occur in both the parasympathetic and sympathetic responses. You don't need to necessarily memorize every single thing that's here, but I think it's good to have a general idea. And the nice thing, I guess, about the nervous system, at least for me when I'm studying it, is like you can really take your own personal experience. So, when you're nervous, how does your body respond? And just think about that when you're asked a question like this. So yeah, it's a hefty diagram, but no need to study it meticulously. I do want to talk about the pathways here. So when you're talking about the actual neural response that occurs in your autonomic nervous system, you have two main components, your preganglionic neuron and your postganglionic neuron. So your preganglionic neuron will get a signal from your central nervous system to release acetylcholine. This applies for both your sympathetic and your parasympathetic nervous systems. And then that acetylcholine is going to attach to or act on nicotinic receptors that are present in your postganglionic neuron. Now, this is something that differs between your sympathetic and parasympathetic systems. For the parasympathetic system, your postganglionic neuron is also going to release acetylcholine, and that acetylcholine acts specifically on muscarinic receptors. And muscarinic receptors are what induce your parasympathetic responses in different tissues. Similarly, in your sympathetic nervous system, you will have norepinephrine released by this postganglionic neuron, and that norepinephrine is going to act specifically on adrenergic receptors in the target tissues. So I don't know if that's something that's super necessary to know for the MCAT, but I think it is definitely a good detail and helps give you a better overview of what's going on here. So there is a distinction between how your autonomic nervous system regulates the sympathetic and parasympathetic systems. Any questions about that? Okay, let's see. So yeah, as you're probably familiar, your nervous system, specifically, especially your autonomic nervous system, is helpful for maintaining homeostasis. And just the overall arching concept of homeostasis, you'll have a sensor, an integrating center, and an effector. So that can refer to homeostasis in your endocrine system. It can refer to your nervous system, whatever you're talking about. And then this functions mainly through feedback. So I think an endocrine example here would be oxytocin during childbirth. So uterine contractions will stimulate your body to produce, or someone who's in childbirth, uterine contractions will cause their body to produce more oxytocin, and that oxytocin will in turn be part of a positive feedback loop, which causes more uterine contractions. And then you have negative feedback, which is a lot more common, I think, in physiology. Just a basic example is blood pressure. So if your blood pressure gets too high, you have, for example, stress receptors that are going to create responses that will help lower that blood pressure in turn. You also have like other types of receptors, I don't know, chemoreceptors for detecting blood pH and stuff like that. And then, yeah, I think just the only other thing I wanted to mention is one of the most important integrating centers is the hypothalamus. So that's the part of your brain that's controlling temperature, heart rate, blood pressure, sleep, all that kind of stuff. So hypothalamus will come up probably a lot when you're studying your neuroendocrine systems. And then, in case I didn't say it before, an effector is just the organ or the tissue that's receiving the information. And that's how that change that's needed to maintain homeostasis is affected. Any questions over this? All right. So we're going to do a bit of a, I mean, this is like an open-ended question. So I just want to like get you guys thinking about how all of this applies to a real world situation. So for example, knowing that that there are different responses in your body that are necessary to maintain homeostasis, how do you think that your body would respond under dehydration conditions? Or how would it adapt to maintain homeostasis? To give you a more illustrative example, like imagine you're walking through a really hot desert and there's no water in sight and you're just sweating buckets and you just are incredibly dehydrated. So how would your body compensate for being in that position? If you're willing to unmute and or type in the chat, please feel free. I'll give you a minute or so. And like no wrong answers, feel free to just guess.
Would it be like maybe sensory, like signals would be sent to the brain?
Yeah, so that's a great start. Yeah, so your sensory input would, for example, be a lot of heat. What can you think about some other sensory inputs? So like that would be one that you're feeling, but I think sensory input here that would trigger a response in your body. I think maybe it's not so obvious, so I'll just go ahead and let you guys know. So one example of sensory input in this situation could be so your body can sense when you have a loss of water because there are receptors for osmolarity in your hypothalamus. So hopefully you're all familiar with the concept of osmolarity. But basically, when the osmolarity of your blood increases, that's because you've lost water, right? And so your hypothalamus is able to sense that. And it's able to trigger a bunch of different responses. So that's about this stage. So can we maybe think of an autonomic, endocrine, and or behavioral response to that. Just like think about what you do when you're dehydrated. Very simple, if you want to think behavioral. Release ADH. Yeah, that's actually correct. Perfect. So yeah, that's your endocrine response here. So your body is going to release ADH or vasopressin, antidiuretic hormone, whatever you want to call it. And that's your endocrine response to prevent water loss through your kidneys or excretion. And also, it also helps to maintain blood pressure because obviously, if you're losing fluid or you're losing water, your blood pressure is going to decrease. And so, by having ADH released, you're going to retain water. That's going to help prevent water loss and maintain blood pressure and maintain osmolarity. Awesome. Perfect. Yeah. And then thoughts on autonomic or behavioral responses?
I mean, for behavior, would you just like sit down, try to like catch your breath? Would it be something like that?
Yeah. And I don't know, just what do you do when you're feeling dehydrated?
Fortunately, I've never like really been dehydrated.
Oh, wow. Okay. Well,
Yeah.
so to prevent from dehydrating, you just rehydrate, right? Yeah, drink water. Exactly. Perfect. So yeah, behavioral response. In this specific situation for dehydration, like talking about osmolarity increasing and all that, your hypothalamus will trigger thirst. And so that's your behavioral response is drinking water. So yeah, stay hydrated. And then autonomic response. So this one maybe is a little less obvious. So in this situation, your body, I mentioned before how your body needs to maintain not only osmolarity, but blood pressure. So an autonomic response here could actually be vasoconstriction. And that would, again, allow not only your body to maintain blood pressure, but it would also decrease the surface area that your like blood is being exposed to. I don't know how to describe this very well, but basically, it would decrease water loss because you wouldn't have as much fluid in your body going close to your surface of your skin, which is really hot. I hope that makes sense. But yes, so you can have autonomic response, vasoconstriction, ADH release, endocrine response, and then behavioral response, thirst, and drinking water to regain homeostasis. Um, and yeah, oftentimes your autonomic response can feed into your endocrine response. So that's just also something to keep in mind is just the bigger picture. And tying it back to your own experiences sometimes is helpful. At least it has been for me whenever I'm studying physiology. So yeah, any questions over that? All right, great job, guys. Let's go on to the rest of our content review. So looking at sensory processing, there are four properties of a stimulus that are necessary for your central nervous system to process it. The first one is location. This is just obviously based on where that sensory neuron is located. But then there's also a concept known as lateral inhibition that you may have come across. So lateral inhibition, I won't go into detail about how it works just because I don't think it's relevant for the MCAT. But basically, what it does is it increases your perception of that thing. So you're more easily able to pinpoint where that sensation is coming from. For example, you're more likely to be able to sense exactly where a stimulus is when it's on your hand or your palm versus when it's on your back. And the reason for this has to do with the neural pathways. So there are primary, secondary, tertiary neurons, and the number of neurons that are present. That usually has to do with how effective or how much lateral inhibition is occurring. If you want me to explain that more clearly, feel free to hang back, but I just won't spend too much time on it because I don't think it's relevant for the MCAT. It's a bit too detailed, I think. And so the second second property is modality. So modality refers to which kind of receptor is being stimulated. Is it a mechanoreceptor? Is it a nociceptor? Is it a thermoreceptor? And then intensity. So intensity is distinguished in specifically by like the frequency of signals or action potentials that are occurring. So if you have a lot of action potentials firing in one go, or like within like 1 second, more neurotransmitter is going to be released by that neuron. And so because more neurotransmitter is being released in a shorter period of time, your brain is going to sense that as a more intense signal. And then lastly is duration. So how long is that stimulus going to be present? And that has to do with, again, the duration of the signal. So either how long one signal lasts, or more specifically, how long the frequencies or how long those action potentials keep firing. And then another concept that we touched on briefly in the homework review, so receptor adaptation. This is the stuff that's, I think, more important than knowing the specific like names, but I don't know, maybe that got changed in the last year since I took it. Basically, you have tonic receptors and phasic receptors. Tonic receptors are slow adapting. Phasic receptors are fast adapting. And so an example of a tonic receptor are like nociceptors, your pain receptors. And those are slow adapting. So think about like if your baby sister pinches you and you feel that pain, it's not going to like be gone as soon as she stops pinching you. It's still there for like a few seconds or even a minute after, depending how hard she pinches. And then phasic receptors are fast adapting. So an example is smell. People tend to go nose blind to things, if you've heard that term. And that's because your olfactory senses, usually your brain is trying to take on new signals as much as possible with smell versus with pain. I mean, from maybe this isn't like asthma, but like from a logic perspective, maybe the pain is something that you need to pay attention to versus smell is something you don't, if that helps you remember it. So yeah, any questions over sensory processing? Okay. So then let's talk about our eye and how that works. So I won't spend too much time on eye anatomy just because I think that's not too difficult to study. I want to go over some of the high yield concepts instead, but definitely know your eye anatomy. So basically, the way that vision works, you have light that will strike photoreceptors in this part of your eye called the fovea. It strikes the photoreceptors directly because there's these overlying neurons here are like pushed aside. And so you'll have your rods and your cones synapsing with bipolar cells, as you can see in this diagram. And one, I think, pathway that's important to understand is the phototransduction that occurs in rods. So basically how light and image gets converted to signals. So rods have discs in them that contain rhodopsin. Rhodopsin is a molecule that gets bleached or activated by light. When that happens, it splits into two parts, opsin and retinol. And this basically will cause a change in the cell, which will decrease cGMP signaling in the cell. So cyclic GMP you probably are familiar with because it's a very, very common signaling molecule. And so usually cGMP levels are high in these rods, but when the rhodopsin becomes activated by light, you have decreased cGMP signaling. The decreased cGMP signaling causes the rods, which are normally releasing glutamate, to stop releasing it. Glutamate is, in this situation, is a neurotransmitter. And what it does is it inhibits, or I guess more specifically, it hyperpolarizes bipolar cells. So it stops bipolar cells from firing. So when you decrease glutamate release from the rods, you end up increasing the activation of bipolar cells. And the reason that this works is actually because bipolar cells don't have your typical action potential. They work on a graded potential. If you're not familiar with that, I do recommend looking, I think maybe at the previous lecture, class 6. Hopefully that was covered then, but if not, feel free to Slack me or just Google it. But yeah, basically bipolar cells function on graded potential. And so they become activated when there is less glutamate being released from your rods. And so these bipolar cells will transmit to ganglion, and that's how all of your visual processing occurs. Any questions over that? Okay. I also just want to mention that lateral inhibition is a concept that comes into play here as well. It occurs via your cells called amacrine cells, and so all it does again is serve to increase or enhance contrast in your vision. So similar to how it helps you pinpoint a stimulus when it comes to sensory processing, it also will just help you increase contrast or pinpoint like clarity in your vision. So that's what lateral inhibition does. All right. And then I believe this is our last slide for content review. This is just over ear and hearing. So once again, please review your ear anatomy. I think that that's pretty basic and important to know when it comes to the MCAT. And again, if you have questions, you can always Slack me or anyone else on our tutoring team. So I just want to go over how signal transduction works in these hair cells that are in your cochlea. So basically what happens from an overarching view, the sound waves and signals go from your cochlea to the cochlear nuclei, which are located in your medulla. And then that signal will travel to your right and left thalamus, and then from there to your right and left auditory cortexes. So the signal transduction in these hair cells occurs when these sound waves cause the hair to bend. When the hair bends, it causes the cells to depolarize, which increases action potential frequency in that neuron. And so these hair cells are actually located on what's known as the basilar membrane. And the basilar membrane is how you have coding for pitch. So whether something is high frequency, low frequency, deep, all that kind of stuff. Oops. Yeah, so how you will figure out what the pitch is of what you're hearing. So the basilar membrane is, it's got an interesting shape. It's sort of thicker on one end and thinner on another end. And I think it might be wider on one end too. And basically what happens is it has a variable sensitivity to frequency. So the hair cells in different locations are going to vibrate or be depolarized by sound waves, depending where on the basilar membrane they're located. And then based on which neurons in which location of that membrane are depolarized, that is how your brain is able to distinguish pitch. Any questions over that? Okay. And so for the sake of time, I did not include slides on taste or olfaction, but if you do have any questions, please feel free to message me or mention it now in the chat or unmute yourself if you have anything you want to go over. But yeah, I think the material that's out there on Khan Academy alone is pretty sufficient and hopefully not too confusing to understand. Okay, so now we can move on to doing a practice passage together. I'm going to drop this link in the chat. And 7 to 8 minutes, I think it's okay if you don't finish, but I think there's only 5 questions, so hopefully it won't take quite as long. So yeah, I'll start the timer and feel free to follow that link that I just dropped in the chat. Oh, and I'll just put up this passage as well because I took screenshots. Okay, let's do another minute, maybe 30 seconds, depending how much time people need. Okay, so hopefully we've all had a chance to look through all the questions. It's okay if you don't have a chance to finish, but just for the sake of time, let's dive into the questions. So the first question we're going to take a look at, what is the most appropriate description of the pathophysiology of multiple sclerosis? So if you'd like, please drop an answer in the chat, or you can unmute yourself if you'd like, whichever you prefer. Okay, yeah, I'm seeing A, and that is the correct answer. Great job. Yeah, so from the passage even alone, even if you don't have any background on multiple sclerosis, there was a sentence saying that it's an immune response against self-myelin cells. So an immune response against the self, that's autoimmune. Perfect. All right, question 2. Which line in figure 1 most likely corresponds to the test group of spinal cords isolated from rats with multiple sclerosis? So again, if you want to type the answer that you put in the chat. Okay, so the answer here was C, and let's just go back to the graph real quick so we can understand why. So the experimental group here, first you have to distinguish what are your controls. So you know you have a positive control and a negative control, and generally those are going to be at your extremes. So in that case, that would be A and D. And then you have to figure out, okay, is my experimental group more similar in characteristics to the negative control or the positive control? And in this situation, it's going to be C. It's more similar to the negative control because your conduction velocity would be severely decreased if those rats have multiple sclerosis, where your myelin sheets are not present. Does that make sense? Any questions?
No, can you repeat that again?
Yeah, of course. So yeah, let me pop back to the graph real quick. Yeah, so as I mentioned, first you want to make sure that you identify which lines here are your positive and your negative controls. And that's because you know that your positive and negative controls are going to be the ones that are most extreme. And so from there you can figure out, okay, either B or C, because you have two experimental groups. B or C, one of them is going to be your experimental group of rats that actually have multiple sclerosis. And so you have to kind of look at the experiment description and the data that might be given to you and decide, is my experimental group going to be more similar to my positive control or my negative control? And so, again, it says here these are synthesized materials as perfect positive and negative controls. One accurately mimics a highly myelinated nerve, and the other is a demyelinated nerve. And so with multiple sclerosis, you have demyelination occurring, which means that your nerve conduction is way slower. And so you know that D is going to be your negative control. And similarly, multiple sclerosis is demyelinated, and so that's going to be closer to your negative control, which in this case is D. Does that help, or do we want to look at the graph a bit more in more detail?
No, that helps. And how did you know that so demyelination, the conduction velocity would be lower?
Yeah, so that's one of those things that hopefully if you have a chance to read through the nervous system, I think maybe this was class 6 or maybe not, but if we didn't cover it, I'll say it now. So your myelin sheath is responsible for increasing the speed of the signal as it travels down your axons. I do think this was covered in class 6. So if you haven't had a chance, maybe you could check that recording. And so basically, if you have demyelination occurring, which is what happens in MS, you're going to have much slower and even attenuated responses or attenuated signals going down those nerves, which is why people with MS slowly lose their ability to control their muscles, voluntary muscles, and even involuntary. And so, yeah, that's the purpose of myelin, is to increase the speed of conduction of your signals through your nerve cells. Yeah, and if you want more detail on that, I'm happy to go through it. I don't know how much is required for the MCAT, but there's a lot that's known about it, and so I can definitely point you to some resources if you'd like later on. Any other questions?
Good, thank you.
Yeah, for sure. Any other questions over this one? Right. If not, we can go to the third one. So this question is more of a standalone. What neural mediated process may be preserved or even heightened after damage to the white matter of the brainstem? So if you're comfortable dropping your answer in the chat, please do. Alrighty, so the answer here is going to be C, your deep tendon or stretch reflex, which is one of those spinal reflexes that we covered earlier. So this goes back to our discussion over the spinal cord anatomy from earlier. And if you'll remember, your spinal cord white matter is responsible for sensory, or specifically, it's responsible for information going to and from your brain. So you kind of have to think about which of these here is not involving your brain. And if you remember, the spinal reflexes can bypass the brain. They go spinal cord and then straight to your motor neuron. The brain may get sensory input, but it doesn't actually need to be functioning for that spinal reflex arc to occur. So that is why you would have this be preserved even after damage to white matter. All these other ones, upper limb coordination, swallowing, and lower limb coordination, require information to and from your brain in order to occur. Does that make sense?
Yes.
Awesome. Thank you. All right. So here's our fourth question. Which feature of the nervous system accurately describes the contents of the gray matter of the brain? Again, feel free to drop your answers in the chat. Okay, yeah, so the answer here is D, abundant neuron cell bodies. So if you'll remember from that diagram that I showed earlier of the spinal cord, you'll have ganglia and nuclei that are present. And so, well, you're not going to have an absence of glial cells, because glial cells are important for a lot of different functions throughout your nervous system. That includes immune protection, nutrition, structural support. So glial cells are pretty much ubiquitous throughout the central nervous system. And then synapses, again, that's something that is literally just required for any sort of neural transmission, because it's that gap between one neuron and the next. So you know that that's ruled out as well. Yeah, and then abundant myelinated nerve axons. So when we're talking about gray matter, we're talking about neuron cell bodies rather than the axons, because the gray matter of the brain is your central nervous system, so that's going to be your cell body, and then your nerve axons tend to be more peripheral. So that's the distinction there. So that's why D is the correct answer. Does that make sense? And do we have any questions?
Can you just go over quickly what was the gray matter and what was the white matter?
Yeah, of course. Let me pop back to that slide, and we can look at it again. Okay, so yeah, so this gray matter is the one that's sort of this darker tan color, and this is your sensory and motor nuclei. So you have your dorsal horn, your ventral horn, and these are just parts of that you can look up to understand their functions a bit better. But overarching view, gray matter is going to be your sensory motor nuclei. And then your white matter is all of this surrounding stuff. And that is the axons that are carrying information to and from the brain, like we talked about before. So with that example of damage to white matter, your spinal reflex arc could still be preserved because your gray matter is intact. And so your dorsal root is where your you would be having like your sensor nuclei coming in, and then your ventral horn is where you would be having your motor nuclei going out. And so this is the gray matter exclusively. And so your spinal reflex tends to be concentrated within this gray matter area. And so white matter is outside, and that's involved in, again, your sensory input to and from the brain. Does that make sense? All right, well, feel free to unmute or chat if you would like more clarification, or I'm happy to go over it towards the end as well. Oh, let's see, there's the chat. The white is the myelin. So white matter is not exclusively myelin. So myelin is what's present around the axons. It's like a myelin sheath around your axons. But the white matter is, yeah, actually, you know what? Maybe I don't know if that's the reason it's colored that way, but yeah, basically the reason that you have white matter is because it's mostly made up of axons. And I guess, yes, technically the color could be from myelin sheets. I don't know if that's like accurate, but it doesn't sound wrong. So yes, because they're axons, yes, technically it's myelin. All right, any other clarification we want on that? Good questions, definitely keep them coming. All right, yeah, if anyone thinks of anything else, we can always come back to it. So this is sort of related to the concept of myelin. What glial cell is involved in saltatory conduction within the central nervous system? So saltatory conduction is what we were talking about earlier about the myelin sheaths. So that fast conduction of a signal from across the axon is called saltatory conduction. It's where the signal is sort of jumping between these nodes of Ranvier is what they're called. So the myelin sheaths have gaps between them that the signal is able to jump through. If there was no myelin sheath, the signal would have to actually travel through every single piece of the axon. But when the myelin sheath is connected, it allows the signal to kind of jump between the gaps and travel a lot faster, and that's called saltatory conduction. So the question here is, what glial cell is involved in saltatory conduction within the central nervous system? So the hint here is what glial cell is related to myelin sheaths, which are involved in saltatory conduction. And also keeping in mind the central nervous system, because there is a difference between central nervous system and peripheral nervous system anatomy. So if you have any guesses, please feel free to drop in the chat. Okay, so the answer here is oligodendrocytes. So hopefully, again, this is okay if you weren't sure about the answer here, because it's definitely one of those more detailed questions, just something that maybe you want to do flashcards to learn and memorize. You have two cells that are listed here that are responsible for producing myelin. Those are your oligodendrocytes and your Schwann cells. Your oligodendrocytes, however, are the ones that are present in your central nervous system. Schwann cells are the ones that are present in the central nervous system. And so I think that's the big, these two are the ones that people would probably lean towards. And then Schwann cells is something someone might pick if they hadn't read central nervous system. So at least, you could narrow it down to these two 50-50. Ependymal cells are in charge of producing cerebrospinal fluid, if I remember correctly. And microglia are immune cells because they have glia at the end. So you can rule those two out. So that's actually the last question from this passage. Do we have any questions or want to go over anything else in more detail? Okay. Well, if anyone wants to hang back, you're more than welcome to. I am going to drop a link in the chat to this survey that we have. We really value everyone's feedback since this is our first time doing like a full prep course, and we really want to be able to make it better for people. So it's only going to take a minute, and if you're willing to give us feedback, we really appreciate it. And I think there's also a raffle involved, so lots of perks. Yeah, so I just dropped that in the chat. If you'd like to click on it and then leave the meeting, feel free. Or if you have any questions, you can stick around. Yeah, thanks for coming. I'm going to end the recording here.